In this video, we build a workflow to quantify bone mineral density and the bone mineral content in a specific section of the femoral cortical bone for a sequence of CT images. The Quick Start button allows to create a new empty generic workflow. In the workflow editor, we add and configure all the required steps. First, we select our CT images. Then, we use a pre-trained machine learning model to segment the femur mask out of the grayscale image. Optionally, we can create a 3D mesh for that mask and visualize it together with the original grayscale image. As a post-processing step, we add connected component labeling and only keep the main component to avoid any mislabeled noise voxels. The next step fits a rotated bounding volume to the segmented mask. This volume of interest covers the entire bone. We can adjust it to cover only a specific section of the bone. At this point, we could add any number of analysis and calculation steps. In this video, we just remove a 30% margin at the start and override the size to a fixed 10% of the total bone length. To avoid resampling the image data, we instead create a binary mask for the volume of interest and use that to mask out the relevant part of the full bone mask. To segment the mineralized bone structure, we now add an automatic thresholding step. It supports various thresholding methods, including Otsu's method. It should only consider the voxels of the bone, so we apply the mask also here, to avoid background noise influencing the result. Starring the plot will show us the histogram once we apply the workflow. We now segment out the cortical bone, based on the filled total bone mask and the thresholded bone result. Another masking step combines this with the specific region mask. A visualization to compare this final evaluation mask with the original grayscale image will help understanding. Now that we have the region to be evaluated, we add a second phantom calibration image to the workflow and assign the calibration from that. The BMD calculation task will calculate both bone mineral density and bone mineral content in the prepared cortical bone region. We select the density calibration to get results in milligram per cubic centimeter units. The finished workflow can now be started. We select the first image and the phantom calibrated reference image. While the machine learning model is busy, we can view the selected image. We can also compare the resulting binary mask to the grayscale image. Or alternatively, we compare the 3D surface mesh to the CT image. It can also be helpful to view the original rotated bounding volume of the entire bone and the adjusted VOI of the 10% region that gets evaluated. Now, we simply add any number of additional images to the workflow, and the same steps will be performed again for all of them. While these images are evaluated, we can view the BMD result and the thresholding histogram. Once all the processing has concluded, we can view and export the table of all BMD and BMC values or display them together with the snapshots.